Good evening, everybody. My name is Mansur 𝗘𝗲𝘀𝗿𝗏. I'm a graduate student at the University of 𝗦𝗶𝘃𝗏𝗰𝗔, and I'm here to present part of our research results titled Innovative Strategies Targeting Escape Pathogens, the Discovery of Potential Bio-Inspired Antimicrobials for hospital-acquired infections. Bacterial infections continue to pose a significant health challenge, leading to increased morbidity and mortality worldwide, particularly in hospital settings. Hospital-acquired infection caused by bacteria remain a global health concern, mainly driven by antibiotic-resistant escape pathogens. Their persistence and resistance significantly hinders treatment success and patient recovery, underscoring the urgent need for new antimicrobial agents. Synthetic compound libraries, often inspired by naturally occurring chemical structures, are composed of lab-designed molecules with broad chemical diversity, making them potential sources for innovative drug candidates. This compound comprises of pharmacophoric groups with previously reported biological activities, including antimicrobial, which may shape them as next-generation therapeutics. Novel chemical entities extracted from our synthetic compound library may exhibit strong antibacterial activity against drug-resistant pathogens, positioning them as promising alternatives for overcoming current antibiotic limitations. The objectives include the evaluation of pharmacological potential of novel antimicrobial compounds derived from a synthetic compound library for their effectiveness against clinically significant bacteria, and also to investigate the molecular interaction between candidate molecule and bacterial targets in order to predict their possible mechanism of actions. A total number of 197 candidate molecules were subjected to in silico virtual screening in order to determine them as oral drug candidates. Initially, the pharmacokinetic property and drug likeness of the compound were analyzed using the Swiss ADME platform, following the 𝗏𝗏𝘃 rule of 5 to assess them as potential oral drug candidates. Followed by energy minimization using the Spartan software in order to determine the stereoelectronic properties of the candidate molecules. Molecular docking analysis was performed using autodock in camera in order to determine the interaction of the candidate molecule with DNA JADIs. Candidate molecules with favorable chemical properties in silico were then tested for the antimicrobial activity against panel of clinically important bacteria. A total number of 72 candidate molecules were tested against members of the ESKAPE bacteria in order to determine the minimum inhibitory concentration and minimum bactericidal concentration in vitro. Initially, the colonies of the tested bacteria were suspended in a test tube containing 𝗏𝗏𝘃𝗏𝗏 broth in order to achieve turbidity that matched the Macfarlane standard 0.5 using the spectrophotometer that was set at 620 nanometer, followed by standardizing the organism suspension to achieve a concentration of 10⁸ colony forming units per ml. All the tested compounds were serially diluted in order to achieve different concentration ranging from 64 to 0.0625 microgram per ml. And then all the inoculated plates were then incubated at 7 degrees centigrade for 20 hours in order to determine the minimum inhibitory concentration. For the minimum bactericidal concentration determination, all the wells that indicate no growth, that means that indicate inhibition, 5 microliter from those wells were spot inoculated onto TSA plates and then further incubated at 37 degrees centigrade for 20 hours in order to determine the minimum bactericidal concentration. The minimum inhibitory concentration was defined as the lowest concentration that inhibited the growth of the bacteria, whereas the minimum bactericidal concentration was determined as the minimum concentration that killed the bacteria. For the molecular docking analysis, the PDB crystal structure of the DNA JADIs was downloaded from the PDB data bank, and then all the molecules that shows inhibition in the in vitro analysis were then extracted from the Spartan software, followed by exporting them into the Chimera software, and then molecular docking analysis was performed in order to determine the interaction of the target protein and the candidate molecules. Then we obtain the following results. Table 1 shows the pharmacokinetic property and stereoelectronic property and molecular docking score of the LBS series. All the LBS series except LBS03 show low gastrointestinal absorption, indicating high level of absorption if taken orally. Whereas for the BBP, that's for the blood-brain barrier permeate, none of the molecules of the LBS was predicted to cross the blood-brain barrier, meaning that they are only able to stay at the peripheral circulation. And going to the enzyme, liver enzyme interaction, all the molecules of the LBS series were predicted not to interact with the liver enzymes except CYP2C9, which was flagged as yes, meaning that there's every possibility that when these candidates finally become drug, their possibility of being metabolized is through the hepatic route, and then of being excreted is through the hepatic route instead of the renal route. For the 𝗘𝗏𝘀𝗃 rule of drug likeness, all the candidate molecules were flagged as yes, meaning that they all obey the 𝗘𝗏𝘀𝗃 rule of 5, which says that candidate molecules must not exceed 500 Dalton in molecular weight. The hydrogen bond acceptor must not exceed 10. The hydrogen bond donor must not exceed 5, and the lipophilicity value must not exceed 5. For the bioavailability score, this is a prediction of the fraction of the compound or of the drug that is able to reach its target without being destroyed. So in this case, 56% of all the molecules of the LBS were predicted to be to reach their target when taken orally. For the HOMO and LUMO properties, all the candidate molecules show favorable HOMO and LUMO values. Actually, the HOMO and LUMO values is a prediction of how stable and how reactive the molecules are. In this case, all the molecules show favorable stability and favorable reactivity. Going to the molecular docking score, LBS01 shows the lowest energy, meaning that it has the highest binding strength when compared to the rest of the compound. Table 2 shows the pharmacokinetic property and stereoelectronic property and molecular docking score of the VFF series. Same as the LBS series, all the VFF series also show high gastrointestinal absorption, whereas in contrast to the LBS series, the VFF series all are flat as yes under the BBBP column, meaning that these molecules are predicted to cross the blood-brain barrier. These molecules are good, could be good molecules for the development of drugs that target central nervous system infection. Going to the liver enzyme interaction, all the columns that are flagged as yes indicate interaction with the liver enzyme, whereas those that are flagged as no are predicted not to interact with the liver enzyme, meaning that all the columns that are flagged as no, meaning that when these molecules later become drugs, their route of excretion would be renal excretion instead of hepatic excretion. Also, the bioavailability score of these molecules are predicted to be pretty good at 85%. The HOMO and LUMO values also are predicted to be favorable. Also, the docking score of the molecules are also predicted to be favorable. Table 3 shows the antibacterial activity of the LBS series against gram-positive and gram-negative bacteria. All the LBS had activity against Staphylococcus aureus, and no activity was recorded against the rest of the gram-positive bacteria, the S. epitabilis and E. faecalis. However, the LBS-01 was able to inhibit the growth of Klebsiella pneumoniae at the highest concentration of 64 microgram per ml. So it means that the LBS 0.1 had a broad spectrum of activity, being able to inhibit both the growth of gram-positive and also gram-negative bacteria. Table 4 shows the antibacterial activity of the VFF series against gram-negative bacteria. Klebsiella pneumoniae was found to be the most susceptible among all the gram-negative bacteria, at concentration ranging from 32 microgram to 64 microgram, with recorded MBC values between 256 microgram per ml to 512 microgram per ml. Figure 5 shows the minimum inhibitory concentration, minimum bactericidal concentration of the Klebsiella pneumoniae. Both compound VFF RJP15 and VFF RJP51 had MBC of 512 microgram per ml against Klebsiella pneumoniae, indicated by these clear zones of the plates. Figure 6 shows the molecular docking results of the of LBS-01 and VFF RJP50. Show the binding of these candidate molecules in the deep pocket of the DNA gyres. This is the LBS-1 in the deep pocket of DNA gyres, whereas this is the VFF RJP50 also in the deep pocket of the DNA gyres. The C shows interaction, hydrogen bonding of the triazole ring with the DNA backbone, and then D indicates hydrogen bonding of the oxygen of the quinone of the VFF RJP50, whereas this structure here is a three-dimensional pose of VFF RJP50 in DNA gyres. In the DNA gyres, where it overlays the original ligand inhibitor, indicating a similar binding pose at the enzyme active site. In conclusion, novel synthetic compounds show promising antibacterial activity against gram-positive and gram-negative bacteria. Compounds exhibit a favorable pharmacokinetic profile, which was supported by their potential as supporting their potential as viable drug candidates. Molecular docking studies indicate strong binding interaction with bacterial DNA gyres, positioning this enzyme as a key therapeutic target. Findings also reinforce the potential of a chemically diverse synthetic compound library as a valuable source for next-generation antibiotics. The results highlight the compound's ability to address the limitation of current antibiotics and also offer solutions against multi-drug-resistant pathogens. The study laid the groundwork for further research, including structural optimization, which may yield even more effective chemical entities. Acknowledgement goes to University 𝗏𝗏𝘀, where I'm currently studying, the 𝗏𝗏𝘀, where the research is being conducted, 𝗏𝗏𝘀𝗶𝗃, and CAPQ for financial support. Thank you.